Dr. Anjali Venkateshwaran, and we will be introducing today's speaker on behalf of Biomere, a preclinical CRO with locations in Boston, San Francisco, and China. Welcome to the webinar series on novel approaches to cell and gene therapies. The speaker for today's webinar is Jeffrey Vick, Chief Business Officer at Chameleon Biosciences. Jeff has more than 25 years of senior management experience in the global biopharmaceutical industry and has particular expertise in gene therapy. He built the business development and IP group at GeneThon, served as the VP of Business Development and IP at the GenCell division of Aventis, and led multiple collaborations with companies like Audentes and GenSite. Jeff has also worked as a venture capitalist where he founded, grew, and sold multiple companies, and also served as the CEO of Silence Therapeutics. He earned an MBA from Stanford University, following a master's degree in chemistry from UC San Diego and a bachelor's degree in chemistry from the University of Virginia. The title of Jeff's talk is Chameleons, Evader Technology, a more potent AAV vector with less immune response. And without further delay, it is my pleasure to introduce Jeff Vick.
Thank you very much, Anjali. It's a great pleasure to be here, and I'd also like to thank Biomere very much for this opportunity to tell you about our Evader technology. The children on our cover slide here are Abby and Belle. They're Niemann-Pick type C patients, and in order to give them a lifelong cure for their disease, it's going to be necessary to have a technology that can be repeat dosed. Chameleon was specifically founded to help patients such as Abby and Belle. As Anjali mentioned, I'm a veteran of the gene therapy industry, as is more so our true founder, Janine Winslow, our Chief Executive Officer. We founded Chameleon with the specific idea of finding a way to improve the current generations of gene therapy and make them more broadly applicable and to be specifically able to treat patients such as Abby and Belle. I'm going to tell you today about our Evader next generation AAV-based gene therapy technology, which we say that's an intelligent immune evasion system. It's also much more potent than first generation AAV vector systems. We are using and leveraging existing production processes, and we maintain serotype specificity, which is to say that the AAV serotype that we're using maintains its characteristics as if it was not put into the Evader technology. This is a platform technology that can be applied across all AAV types, and it has very strong intellectual property associated with it. I'm going to show you today in vivo data, which will show the significant advantages that Evader has versus normal AAV technology. And we're going to do that in a mouse model of human factor IX, using AAV8. We're using this data because it has been published by other groups with normal AAV and by Dr. Casey McGuire, who I will mention more, for enveloped AAV technology. Using this system, we will show that we have 20 times more transgene expression than AAV alone, 10 times increased gene delivery to the liver. We have reduced antibody response, and in these specific experiments, we're not going to show it because these are focused on antibodies, but we've also demonstrated that we have a greatly reduced T-cell response in our human in vitro data. Finally, we have taken this technology and we have put it into a manufacturing system which is scalable and which is resulting in a process which we can move forward to commercialization. Gene therapy has shown some stunning successes today. In severe hemophilia, both type A and type B hemophilia, in spinal muscle atrophy with the breakthrough product Zolgensma of Avexis, in congenital blindness with the Luxturna product of Spark, and spectacular data in the X-linked myotubular myopathy trial of Audentes. However, these first generation gene therapies suffer from a number of problems and limitations, which will limit the patient types that they can treat, and also will limit the ability to provide a lifelong durable cure for patients with certain diseases. One of these problems is simply based on the fact that the immune response limits the number of treatable patients. There's currently a one-dose limit with first-generation gene therapy vectors. This is because, like, we use AAV is a virus, and like any other virus, when it is given to a human patient, it sets up a very strong immune response, especially at the extremely high doses of viral load, which are given in AAV gene therapies. It is essentially treated like a vaccine, and the human body will set up an immune response against that gene therapy treatment. So you cannot go back and treat a second time with the same capsid serotype. The data on the right are from Amit Nathwani, and he is looking at immune response after treatment with gene therapy, in this case AAV8. The second problem, which is related to the first problem, but somewhat different, is that the gene therapy delivery systems, the AAV viruses, which are used to deliver first-generation gene therapy products, come from naturally circulating viruses. So there's a significant chance that an adult patient in particular can have been exposed to the AAV serotype in just the environmental circulation prior to entering the clinic for gene therapy treatment. This can be a particular problem, especially in adults, and it depends on which serotype we're talking about. It can be more or less depending on the serotype, but in general, up to about 50% of adults can't be treated based on prior exposure to AAV if they have neutralizing antibodies against AAV. And this is a typical entry criteria for entering into a gene therapy clinical trial. If you have neutralizing antibodies above a certain level, then you're simply excluded from treatment in that trial. The third problem that I want to talk about is that a single dose is not always durable. The data on the right are from BioMarin. We're looking at their trial in hemophilia A, where they're looking at factor VIII activity over time following a single dose of gene therapy in adult hemophiliacs. They are definitely seeing a decline in factor VIII levels over time. And if one extrapolates this out, they believe that in approximately 8 years or so, it will be necessary to give these adult patients a second dose. The problem is that with the current gene therapy treatments, that's not possible because of the strong immune response that has been set up against the capsid in the first gene therapy treatment. This is also very significant for children. And as a result, for example, in hemophilia, because there are already enzyme replacement therapies, children are not allowed to be treated in the current gene therapy trials for hemophilia. And the reason for this is that even if the effect did last for a very long period of time in a non-dividing cell or in a cell such as the liver, which is where the hemophilia gene therapy treatment is targeting, what will happen is that after a first dose of gene therapy AAV treatment in a child, a certain proportion of liver cells will be having the replacement gene to affect the disease. However, as the child grows and their liver reproduces and the liver expands in size, the new liver cells will not have the replacement gene. And so even if the gene therapy stays in the initially transfected cells, over time, the effect will wear off and will wash off. So currently, gene therapy treatments are not appropriate for children where there's an alternative. Of course, in tissues which are completely not dividing or in very life-threatening diseases where there's no other alternative, such as the SMA trials, then children are able to be treated. But in the current hemophilia trials, that is not the case. So there are a number of clinical challenges which are experienced with gene therapy treatments. And I first want to look on the part of the slide to the right, and we will go over time. So the first thing is pre-existing immunity. So as we mentioned, the AAV serotypes circulate naturally in the normal environment, and so there is a chance, and that chance rises with time because there is longer time to be exposed to native type virus, that the patient will arrive in the clinic with neutralizing antibodies that will prevent the first dose of a gene therapy treatment. Also T-cell immunity, and this can simply preclude a patient from having a dose in the first place. The second problem is what happens after the gene therapy treatment is given, and that is that there is an innate immunity to the vector, to the AAV virus, which is delivering the gene therapy, which can result in acute toxicities. And this problem is more and more important the higher the dose of vector virus is given. There have been recently a number of 2 deaths in the trials of the Audentes myotubular product, which I might mention is showing spectacular clinical results in patients. But nonetheless, on 2 patients, which were larger patients, older patients, and therefore received a very much higher dose of the vector based on the fact that it's dosed by weight, in those patients which received a very, very high level of vector copies, they had acute problems which led to their death. The third problem is adaptive immunity. Even if the patient gets beyond the acute toxicity, the patient will set up an immune response, both a high titer neutralizing antibody response against the capsid, as well as a cytotoxic T cell response against the capsid. And there also can be immune responses against the transgene itself. And so it is this third problem which is preventing repeat dosing of current first generation gene therapy vectors. Looking at that another way, on the left, this is related to 3 different things. First of all, current first generation AAV vectors are low potency, which means that very high viral loads have to be given to achieve a certain therapeutic effect. The higher the dose of the capsid, the higher the immune response is, which makes sense. Interestingly enough, this low potency and the higher immune response also directly correlates to lower efficacy of the transgene itself, and it can cause acute toxicity. As a result of the limit of 1 dose per patient per lifetime, current AAV gene therapies are given essentially at the highest dose that has been seen in previous clinical trials to be tolerated, and then titrated based on weight, because you can't go back and titrate the dose by giving a second dose. Our products are much more potent than first generation gene therapy vectors, and therefore we can give a lower dose in terms of both the vector copies that we are giving to achieve the same effect, and also in terms of, importantly, the viral load that we're giving, which by itself lowers the immune response to the product that we are using. The second problem is this pre-existing immunity that we mentioned, the neutralizing antibodies and T cells which exist prior to the first exposure of gene therapy, based on the fact that the patient can have received or can have been exposed to AAV of a close enough serotype to have immune reaction. The third problem is the immune response after the gene therapy dose, which prevents repeat dosing. And the Chameleon evader technology that we're talking about is designed to address all 3 of these problems. This addressing of these clinical problems then also confers multiple market advantages to our product. First of all, it's 20 times more potent, which means that we can have increased gene delivery and therapeutic gene expression with lower viral dosage. We maintain the tissue target specificity. What I mean by that is if we are using an AAV8 in the evader system, it will have the same tissue tropism as normal AAV8 without the evader serotype. It will be more infectious, it will deliver more transgene, but it will have the same tissue target specificity. Thirdly, we have resistance to neutralizing antibodies, which means that we can broaden the patient population and treat patients that otherwise would be excluded from clinical trials based on the fact that they have neutralizing antibodies when they present in the clinic. And then finally, and importantly, we are going after repeat dosing. We are doing that by using our technology where we have immune suppressors masking the particles that we're using from the immune system, and I'll talk about that in detail. I'm not going to go through all the benefits of repeat dosing, other than the fact that we can give a second dose as required to treat a patient such as Abby and Bell, that after a number of years would require a repeat dose to have their gene therapy continue to be effective. But there are other benefits as well. As I mentioned, current first-generation gene therapies are given at essentially the maximum dose that could be given to a patient without having problems from overload of virus. And therefore, you can't titrate the dose. If we can have a system which is repeat dosable, then we can start with a smaller dose and we can give more doses over time, if required, to the patient to titrate up the level of the gene therapy. And we can start to have a product which is looking more like a traditional small molecule product, where you titrate the dose based on the specific requirements of that patient, rather than doing a one-size-fits-all kind of approach. So how do we do that? So let me talk specifically about the Evader platform technology. We start with a standard AAV of any serotype. This can be the standard serotypes that people are widely using right now, such as AAV8 or AAV9, or it could be the next generation serotypes, which people are working on to achieve different target specificity, to have better transduction, or simply to avoid pre-existing antibodies, because the engineered capsids are ones that are not normally circulating in the native environment. We can work with any of those kinds of serotypes, and we start with exactly the same thing that is being used in terms of the transgene inside as well. There's no differences there. What makes the difference for the Evader technology is, first of all, an enveloped AAV lipid bilayer that is on the outside of these particles. Our co-founder, Dr. Casey McGuire at Harvard University, who gave a previous Biomir seminar, has invented this technology, which he calls exo-AAV for exosomal AAV, and which is also called enveloped AAV. The enveloped AAV, the exo-AAV, by itself confers certain features and benefits which are very advantageous. Number one, that lipid bilayer by itself improves the gene delivery. Secondly, it also provides some resistance to pre-existing neutralizing antibodies. So we started with that technology, but we then built upon that. We actually thought about these particles as if they were something like lentivirus, which natively has an envelope around it. And in fact, what Dr. McGuire discovered is that by changing the way that you purify AAV particles, but using the same producer cells, the HEK293 cells or other cells that you use to produce AAV, you can produce an enveloped AAV. And once you have that envelope on them, like a lentivirus, where you can pseudotype the lentivirus to put proteins on the surface of that envelope, our founder, Janine Winslow, thought that she could do the same thing. And it turns out that that is exactly the case. So the Evader technology, first of all, has CTLA-4 expressed on the surface of this envelope lipid bilayer. The CTLA-4 reduces both T and B cell activation, and it helps to mask the particles from the immune system, and is one of the important features towards allowing repeat dosing and immune system reduction based on our particles. The second factor that we're using is PD-L1. This is also reducing T cell activation, inducing tolerance, and helping to allow repeat dosing. These features together, the enveloped lipid bilayer, the CTLA-4 expressed on the surface, and the PD-L1 together are what we call the Evader technology. I might mention that the molecules that we're talking about here, CTLA-4 and PD-L1, are very well characterized, including in humans. CTLA-4 itself is available in a soluble form and is used for treatment of both rheumatoid arthritis and transplant rejection. And it's known it's well tolerated in humans, and it's known very well to have these immune reduction properties that I've talked about. PD-L1 is actually quite interesting. There has been quite a renaissance in immuno-oncology over the last years, as we're all aware. And one of the breakthrough drugs, the so-called checkpoint inhibitors, is a drug called PD-1. PD-1, in fact, is the inhibitor of PD-L1. PD-L1 is the ligand. It has been shown that tumor cells will express exosomes, will shed exosomes with PD-L1 on their surface. The PD-L1 on their surface will shield those tumors from the human immune system, and that's why then the checkpoint inhibitors, which reduce this effect, can have such beneficial effects in cancer patients. So we're taking advantage of the knowledge that has been gained over many years to design a better gene therapy system. And we call this system the Evader platform technology. Dr. Casey McGuire and other laboratories around the world who have repeated this data have shown that enveloped AAV is much more potent than standard AAV. I'm going to focus on AAV8 for the rest of this presentation, and I'm going to focus on this mouse model of hemophilia B showing factor IX expression, but this has been demonstrated for many other AAV serotypes and for many other conditions and transgenes as well. So first of all, enveloped AAV is much more potent than standard AAV. So if you look on this and on the far right, first of all, enveloped AAV will result in higher numbers of gene copies delivered per cell. Secondly, on the far left, this translates to more factor IX production and also factor IX activity. So that's important as well because it's not just the protein that's produced, but it's also effective protein. Secondly, the enveloped AAV is also helpful in overcoming the presence of neutralizing antibodies because of this lipid shield around it. So the in vivo infectivity in the presence of neutralizing antibodies is shown here, and it's much more effective than simply standard AAV itself. However, the enveloped AAV8 does not do anything to address the induced immune response. Enveloped AAV8 will still result in a very strong immune response against the capsid and therefore is not appropriate for repeat dosing. That's why we developed the Chameleon evader technology, which I described to you. So in this experiment, in the same model, which we're doing because people have published on this with standard AAV and enveloped AAV. But in this experiment, we've actually used them directly. So this is not comparison with literature. This is actually a direct experiment where all were tested together. We first of all show that in terms of factor IX gene delivery, we are having the equivalent numbers of copies per cell as the enveloped AAV. So if you look here on the left, the enveloped and the evader AAV both result in much higher levels of gene copy numbers per cell when the dose is normalized. So that's repeated from what the literature previously says. But what we also see is that not only do we see higher expression based on the number of gene copies, but it's even better than that. We're actually seeing a much more potent delivery of factor IX protein than even the enveloped AAV is showing. So even though the enveloped AAV and the evader AAV result in essentially the same number of gene copies per cell, the evader of AAV product is showing much more protein production, factor IX expression production. We believe that this is an effect based on lowered inflammation. It has been shown that in publications that inflammation of the liver, which is the result of gene therapy treatment, can result in reduced transgene expression. And we believe that by reducing the immune response, reducing the inflammation, we are seeing better expression of factor IX, even when the gene copy number is normalized relative to the enveloped AAV. We also have shown that we can lower the immune response in terms of antibody production. So here we have administered in the same factor IX mouse model, standard AAV, enveloped AAV, or evader AAV. And we have dramatically reduced the immune response after 2 doses of treatment by the evader product. I might mention that this experiment was using relatively dirty preparations, and as we clean up the material, things are looking even better in terms of transduction and in terms of expression, and we believe in terms of lowering the immune response. So we believe that we have a strategy to ensure successful repeat dosing based on this. The potential clinical benefits on the right are, first of all, we can look at doing a lower dose with the same efficacy. That lower dose, based in terms of gene copy numbers and in terms of viral load, results in, even if we didn't have the immune masking effect, just by having a lower dose, you lower the immune response just for that reason. Secondly, we're looking at having higher therapeutic protein levels possible than with other forms of gene therapy. We're looking at a decrease in T-cell mediated inflammation, and we're looking at the potential to redose when needed. These things are enabled by the fact that we have a product which is more potent, which reduces T-cell activation and reduces B-cell activation. We have very strong intellectual property. So we are building on the technology of Casey McGuire, this exo AAV, for which patents have already issued. In addition, we have filed intellectual property filings on composition of matter, method of production, method of use for the Evader technology as well. And it looks very promising for this to be allowed because it does not seem to be anticipated whatsoever in the literature or by other companies or other investigators. Our portfolio at this moment is being developed for certain products internally, but we will then broaden that up and partner it over time. So initially right now, we have a portfolio where we're focused on orphan diseases, which will give us a very rapid path to clinical data. And in these cases, predefined clinical endpoints, because people have worked on these diseases before, either with gene therapy treatments or with other forms of treatment. So something is known about these diseases. MinG on the left is a rare genetic mitochondrial disease which affects a few number of patients, but patients which will require repeat dosing. And it's one where we can get very rapidly into the clinic, and there's a very significant unmet medical need, and that can move very quickly. Niemann-Pick type C is a horrible disease for which there is no gene therapy treatment at the moment. Patients are waiting for such a treatment, and patients such as Abby and Bell, which I mentioned at the beginning, will require repeat dosing in order for them to have a lifelong durable benefit from a gene therapy treatment. And that is a significant market as well. Finally and importantly, we're working on severe hemophilia, hemophilia A and B. This is one of, if not the largest segment of rare diseases. And people sometimes ask us, well, why are you working on hemophilia, given that there are so many other companies working on hemophilia with gene therapy treatment specifically? And the answer is that we are actually not in a competitive space. The current therapies which are being developed for both hemophilia A and for hemophilia B are targeted at adults who do not have pre-existing immunity. We, in fact, are able to go after those other patient populations and dramatically expand the market. It's potentially a $24 billion market for hemophilia A and B combined. Once we have clinical data, and perhaps even before that, we're open to partnership opportunities right now. This is a platform technology for which we can offer line extensions to the current first-generation gene therapy companies. So as I mentioned before, we can work with any capsid and any payload any capsid of AAV and any transgene payload. We're using the same producer cells that everybody else is already using. We're using the same production plasmids. You don't have to re-engineer anything in terms of the gene constructs in order to use the Evader technology. We simply produce it slightly differently and purify it somewhat differently. We can also work with other payloads than just traditional first-generation gene therapy, such as CRISPR-Cas9 technology. So there are lots of opportunities to provide companies who either have a product in the market or who are developing a product in clinical trials or who would like to think about a superior product before they even enter into clinical trials to partner with Chameleon to improve their product offering. As an example of what this can result in terms of market benefit, the current market for hemophilia, this is based on data from the World Federation of Hemophilia report, which is very detailed data. It's approximately a $24 billion market. However, in the United States and Europe alone, not counting the rest of the world. However, the current therapies which are being developed only treat adults without neutralizing antibodies and cannot treat children. So that limits the market to about $12 billion, which is still an extremely substantial market and the obvious reason why many companies are going after that. We want to, however, treat more patients. Using the Chameleon technology, we can increase that market quite dramatically. We can treat more patients with pre-existing immunity, which means we can treat adults with pre-existing immunity, and we can start to think about treating children if we can have a technology which is repeat dosable. The final thing I want to mention, and I'm not going to go through extensive data on this, but we're leveraging existing production technology. The evader production technology is leveraging what we already know about AAV and lentivirus GMP production technology. Our founder, Janine Winslow, has been involved in manufacturing and CMC of both AAV and lentivirus products, and we've leveraged that technology. Essentially, on the bottom left, we have a producer cell, which typically would be an HEK 293 cell, on which we have overexpressed on the surface of that cell our immune signaling molecules. We produce the AAV in that cell and it buds off like exosomes. But the difference between what we're doing and what our co-founder Casey McGuire is doing, where the exosomes would bud off, is that our enveloped AAV particles express the immune masking molecules on the surface of them because they were expressed on the surface of the producer cells. We then are using the same production starting materials, as I mentioned, the same transgenes if we wanted to port over a first generation product and make a second generation product, and the same producer cells. We then harvest that material and we purify it much like we would purify AAV, and we'd formulate it much like we would formulate current products of AAV. And then we will have a release and stability testing like we would for AAV, with some additional release and purity, stability, quality measurements based on what we know from lentivirus technology as well, since AAV that we're using in this kind of an evader system is an enveloped virus system. So in conclusion, the Chameleon evader technology offers a superior gene therapy platform. We are reducing the immune reaction to the product, which we are also resulting in superior gene delivery, both in terms of copy number, but very importantly also in terms of transgene expression. This is a broad platform that's applicable for any type of AAV gene therapy using any AAV serotype, whether it's the traditional ones or the ones that are being engineered now. And these benefits translate into very substantial advantages in the clinic and therefore also in the marketplace. For example, in the case of hemophilia, we can double the existing market from a 12 billion opportunity to approximately a 24 billion opportunity. The pipeline that we're working on right now is for hemophilia, Niemann-Pick type C, and MNGI, which is the mitochondrial disease. But we would welcome inquiries from any company or from any investigator that thinks that their research could benefit from what we're doing. This is all protected by a very strong patent position, which means that the technology is also appropriate for commercialization and for putting into the marketplace. I'd like to thank you again very much for your attention, and would be happy to answer any questions that you might have.
Thank you, Jeff, for a wonderful presentation. And it's so exciting to see how the evader technology has evolved. So, one of the, I think, holy grails of gene therapy is delivery. So whether it's systemic versus local, that's, I think, one of the biggest areas across different diseases. So I don't know if you had, sorry if I missed it, but how is the enveloped AAV delivered? Is it intravenously? Do you know?
Yes, so we can deliver the evader technology in a number of different ways. Primarily, we're going after systemic applications at the moment. And for systemic applications, we're looking at systemic delivery. However, it's also appropriate for any of the other forms of delivery that the traditional AAV is being used for. So for example, it could be administered in the eye potentially. And although, for a long period of time, it was thought that the eye was a immunoprivileged site and was not resulting in immune reaction, what we're actually seeing in scientific conferences is that there is starting to be immune reaction against AAV given in the eye. It could also be potentially administered intrathecally for products where the CNS effect is important, or other places as well. So really any place that you could give a traditional AAV, we're looking at that ability as well. But we're initially focusing on systemic applications because those are the most life-threatening areas where we can have the maximum benefit, and we really want to help patients.
I agree. And I definitely, it makes it a lot easier to administer, especially if you're going to the liver or something like that, to do a systemic administration, especially in children, as opposed to doing a much more invasive delivery method. So I think that is very exciting, because one of the issues, and you had mentioned this, and it is that, a lot of times if you have to do systemic unenveloped virus, you've got to give a very high viral dose. And that just has its own issues and complications. Whereas with the Evader technology, you're still able to keep the dose relatively manageable, but are able to overcome the antiviral response, which is I think a huge deal.
Absolutely. Let me even just make another comment on that. So, I and my founders have been involved in a number of gene therapies. And so, for example, the Audentes X-linked myotubular myopathy therapy and others. And, oftentimes when the systemic gene therapy is given, even to a child, the physician will have to administer on the order of 100 vials of product. I mean,
Yeah.
that's a logistical problem.
Yeah.
It complexifies everything, and also that there's manufacturing issues and just supporting the
Yes.
scale of manufacturing. If this translates and we can reduce the dose by a factor of 10 or 20 or even more, it will reduce both the numbers of vials required, and it should also dramatically reduce the, even forgetting about the, the immune masking for a moment, it should reduce the side effects which are associated with huge viral load. So we're really excited by the potential to lower the amount of virus that we're given.
And this has implications across, I think, almost every disease where gene therapy is investigated. So that was my next question is a little bit more forward looking. And I know a lot of the fantastic work is with AAV, which makes a ton of sense because it's such a well-studied viral vector. But I guess I think the obvious question is, are there plans to expand beyond AAV and maybe look at, and if there are, what would be the limitations? Would it be a certain, because AAV being small, that fits in very nicely into envelopes, but is Chameleon thinking of expanding beyond AAV?
So there's a short-term and a longer-term answer to this question. In the short term, we're a small, focused company that needs to demonstrate what we need to demonstrate to move the technology forward. However, we can look at doing some partnerships early on to expand into other areas. Certainly longer down the line, the goal of the founders of this company is to broaden the gene therapy to the patients that we can cure, really, to help cure patients for lifelong cure. And we are focused right now on immunosuppression using these molecules, but we also could use immune activation, and we could go after oncology, for example. So for example, for oncolytic viruses, for example, that's an obvious application that we would be interested to talk with companies about collaborations in the future. And then beyond that, lentivirus is not typically used systemically yet, but there have been some probe experiments with lentivirus, and so this could be applied to lentiviral technology as well and other applications. So the short-term answer is that we have to focus on what we're focused on, which is our pipeline and demonstrating the benefits. But once we've succeeded in doing that, then yes, indeed, we will look to broaden the technology to other areas.
That's good to hear, because I think there's so much implication, because this is answering such a key bottleneck or limitation in applying, whether it's oncolytic viruses, whether it's viral vectors in rare diseases, whatever it is. I think there is always that challenge and that issue. And for example, off the top of my head, when you mentioned oncolytic virus, I was thinking, oh, it'd be so cool if they could envelope HSV1, which has been used a lot.
Yes, absolutely.
And so I think there's just tremendous potential. So I can definitely say that this technology is really going to, once proven and in the clinic and things like that, I think it has tremendous potential to really change the way, gene therapy is delivered.
Thank you. And we would like to see how this is used with other, breakthrough technologies.
Yeah.
We're not a company that says we're going to do everything by ourselves and this technology will solve all problems. We would love to work with companies that have next generation capsids for different targeting and for other purposes. We are very open to stay on the forefront of what's happening in gene therapy. We think that we have a wonderful offering, and that we think that this can benefit many other kinds of technologies and companies.
Definitely a very, very elegant approach, I must say.
Thank you.
So with that, thank you, Jeff, for a wonderful educational presentation. If you would like to learn more about Chameleon Biosciences and how the technology can be applied to several diseases, including hemophilia, like Jeff presented, Niemann-Pick type C, that Jeff had mentioned, as well as other diseases like Duchenne muscular dystrophy, Pompe disease, etc., please visit chameleonbiosci.com for more information. And also please feel free to check out Dr. Casey McGuire's webinar that Jeff had referenced, also available on the Biomere website. So with that, thank you, Jeff, again, and it's been a pleasure listening to your wonderful presentation.
Thank you again, Anjali, and thank you very much, Biomir. This was a wonderful opportunity. We certainly appreciate being able to tell our story.